specific organisms, looking for a drug for a specific organism to target one particular condition. I think that's probably feasible in the future. Another way that we can use this assay is also to look at cytotoxicity. So one of the lead compounds that we have for Nocardia farcinica shows no cytotoxic activity against a range of human cell lines, shows some mycobacterial activity and some trypanosome activity, but it shows a lot of activity against farcinica. So these are being developed currently. So in the last part of the talk, I'd really like to go back to my first love, which is Streptomyces. It's a large genus, generally aerobic soil saprophytes, but as we all know in the audience, these are organisms that cause this debilitating condition that we've heard so much about in the last couple of days. They're very famous for their extensive production of secondary metabolites. They have large genomes by bacterial standards. They have interesting and complex developmental life cycles, which are illustrated below. We know that there are pathogens of plants within these organisms, but also pathogens of humans. How am I doing for time? Okay. Doing okay. So one of the ways that we would like to try and understand this organism is through genome sequencing, which then, as I've alluded to with the Nocardia story, is to try and then develop tools to study the pathogenicity of these organisms and try and understand their ecology and their biogeography. So we need this deeper understanding of why somaliensis and pseudonensis are causing diseases, but we really need to think carefully about the strains that we begin to sequence, because obviously an old strain that's been passaged through culture on agar plates for a long time is likely to be selected against the kinds of factors that are likely to induce disease causality in the host. So we took the approach of looking across the whole grouping of the Streptomyces and also looking at where the pathogenic Streptomyces lay within this tree. So we had a choice of strains, and we knew that there was some variation within the isolates. We also have the powerful tool of comparative genomics. Because of the biotechnological interest in Streptomyces, there are about 28 other whole Streptomyces genomes known. So this gives us a tremendous resource in being able to compare our pathogen sequence against known non-pathogenic strains. So that immediately makes it easy to identify regions of the genome that are different and therefore are likely to be encoding our genes for pathogenicity. This will speed up the identification of virulence factors and will also allow us to identify specific genomic features of our strains of interest. So in terms of choosing strains, we had access to several isolates that all largely came from Ahmed's lab, and also the type strain of Streptomyces somaliensis, which you can see illustrated here. Okay, so you get this clade from a paper that Ahmed and Mike published, and I guess some of you probably know Erica Quintana, who spoke at the last meeting, or maybe 2002 meeting. So this is the type strain, and then this is the new isolate that was named Streptomyces pseudonensis. And then we also have an isolate here, SD509, which is an isolate from Ahmed's lab of Streptomyces somaliensis. Now, whether this is a different organism, we don't know. But what we decided to do was sequence these three strains. So we've got the type strain of somaliensis, the type strain of pseudonensis, and then potentially a new isolate of somaliensis, which will allow us to compare a strain that's been passaged through non-host culture, and also a strain that's relatively recently been isolated from the host. So we took a new sequencing strategy. Previously, people would make genomic libraries, and then they would use classic Sanger type sequencing sequence the libraries, and then you would align lots of these cosmids, and then try and piece the genome together like a very big AGTC jigsaw. But we wanted to use a more novel and more rapid technique, so we used the next generation sequencing, which is based on fluorescent labeling of your DNA, and you get a flash of fluorescence as you wash over your bases within the sequencing reaction, and you can extend your knowledge of the sequence from that. So in Strathclyde, we grew up the organisms, and we prepared DNA from all of the strains. And I think an interesting aside here, that traditionally these organisms have been very difficult to culture. And the media that have been used have been very complicated, difficult to make, and not necessarily easily accessible for all of the components. I do a lot of work in my lab on Corynebacterium diphtheriae, and we spend a lot of time making brain heart infusion agar to grow these organisms. And one day I had a few plates left over, and a few plates of Streptomyces somaliensis, so I thought I'll streak it out onto this medium and see what happens. And traditionally we would think of Streptomyces somaliensis as very slow growing, taking maybe 5 or 6 days to really come up on the plates. And 24, 30 hours later, we had extensive growth on the brain heart infusion plates. So I think we've got a really good way of cultivating these organisms now that's very quick. It also grows very nicely in liquid culture, very dispersed, which makes it easier to extract DNA, whether it's for genome sequencing, or for cloning purposes, or for MLST purposes. So with our collaborators in Taiwan at the Yangming Genome Center, we sent off our DNA over to Yangming, and they prepared the library through fractionating the whole genomic DNA. They then ligate an adapter, which allows you to then tether your DNA sequence to a chip, which then allows you to use this next generation Illumina Selecta sequencing. And we then got our data back a few months later, and we had 500 megabases of genome sequence. So we had quite a lot of depth, so there was a lot of sequences for all of the genome, which made assembling of the contigs, so the contiguous regions of the genome, a little bit easier. So from next generation sequencing, you get reads of about 50 base pairs, so you then need to use some complicated algorithms and bioinformatics software to assemble these contigs, which I don't pretend to understand, but our collaborators have a lot of experience in this, and they were able to assemble some of the short reads, which were then sent to me, and I was able to assemble them into larger contigs. Again, it's like piecing a jigsaw together. It's time consuming, but it's not particularly difficult. And then we were able to start the hard work, which is annotating the genome, which is really assigning function to all of the coding sequences that we can find within the genome. And then at a later stage, we'll then need to start filling in the gaps, because obviously there will be gaps within each of these contigs that we need to cover to get the complete genome sequence. So I think it's very exciting that I can talk about some of the preliminary analysis that we've done here within the home of Mycetoma. So this is literally fresh out of the sequencing machine. We've had this data for probably 3 weeks, 3.5 weeks now. So I think you guys are hearing this for the first time. Nobody else knows this other than probably me and Mike that I've discussed it with. So we began with the preliminary analysis. We assigned the open reading frame, so it's a way of identifying the genes. We used a piece of software that is freely available, again, from the National Bioinformatics Institute in the US, called Glimmer. And this uses an algorithm and basically looks for start codons and stop codons within a gene sequence, and then assigns this as an open reading frame set by your parameters. So we chose the parameter of 100 amino acid proteins. Generally, you don't tend to see many proteins less than 100 amino acids. And then we can go back at a later point, once we've assigned all of these genes, and look at gaps within the genome annotation and fill in the bits and pieces that we need to. And then we're using a piece of software called Artemis, again, It's a very powerful piece of software, but it's freely available from the Sanger Institute in Cambridge. And this tool runs on a Unix platform, but there are also platforms that allow you to use it on a Windows machine. And we can assemble all our contigs, as you can see here, and we've begun the process of annotation that you can see up here. This is my small effort in the last couple of weeks at beginning to annotate the genome. So we still have quite a long way to go. It's early days. But what we can do from these contigs is infer through blast searching and also looking at the genome as a whole, some interesting properties of this organism. So this is a screenshot of Artemis, which demonstrates to you how you can look at this sequence. So you have the positive and negative strand of DNA here, okay, from the sequencing, and then you have the translation of the DNA into protein sequences in 3 frames above and below this. The software also automatically calls all of the stop codons, which you can see here as black lines within the translated sequence. And then this allows you to find regions that are likely to be open reading frames, which are indicated by the blue. So these are ones that I've already annotated, just to show you the process. Then you can take these sequences once you've found them, and then you can run a blast search, so a bioinformatics search, to look for sequences within the database that are homologous to this, so you can then try and infer some biological function for these genes. So the slow process has started in terms of this, as I said before. But you can also do interesting things like this is a GC frame plot. So it looks at the codons within the DNA and allows you to predict regions of the genome that are likely to encode genes. And you can see here that the frame plot increases in this blue line and extends along and then drops down, and this equates to this coding sequence here, so you can check the validity of your glimmer predictions by using the frame plot. Also, something that Streptomyces biologists have become obsessed with over the years is GC content within their organism, and this allows you to plot the overall GC content within an organism. And I think Mike will mention a little bit more about this in terms of identifying virulence factors in his talk, so I won't steal his thunder. So we're proceeding slowly. We've aligned into some big super contigs of maybe half a megabase, and we can now start to use that information at the edges of the contigs to find the contigs elsewhere in the genome that are likely to overlap with them. And this has been advanced by our knowledge of other Streptomyces genomes, because we have a knowledge of synteny. So this is regions of the genome that are largely similar across a lot of strains. So you tend to see a core genome within organisms. This allows you to infer some degree of synteny, which makes the annotation and the assembly of these contigs much easier. So really cutting to the chase, and the thing that will interest you all is what makes Streptomyces somaliensis interesting and likely to be a pathogen. Well, it has a smaller genome than all of the other Streptomyces that have been sequenced so far, and this is a trend that you tend to see in a lot of pathogens. Adaptation to living in the host, which is a much more stable environment for pathogens, they tend to lose genetic material, so there is always a trend to a smaller genome in a pathogen. The sort of exemplifying case of this is Mycobacterium leprae, which is a clonal derivative of Mycobacterium tuberculosis, and it's undergone massive gene decay. The genome's about 50% of the size of tuberculosis in terms of coding sequence. It encodes about 40 sigma factors, so these are the part of the RNA polymerase subunit that encodes specificity for transcription. Again, most organisms, most normal bacterial organisms have between 1 and 10 sigma factors. Streptomyces is unusual in this point, and our favorite pet genetic Streptomyces coelicolor, has 65 of these sigma factors. So in somaliensis, I've only been able to identify 40 sigma factors so far, so either there's 25 lying in the gaps that we haven't yet sequenced, or there's less sigma factor activity within this organism, which again suggests that its niche is likely to be constrained compared to the organisms that live in the soil all the time. I've identified these TAP-C and TAP-L proteins, which are involved in stabilizing the terminal inverted repeats at the ends of linear genomes in Streptomyces. So this allows us to conclude that Streptomyces somaliensis has a linear genome, which is very unusual amongst bacteria, but not unusual amongst the Streptomyces species. So again, it behaves like a Streptomyces in terms of its genome structure, but the structure is much smaller. There are multiple non-ribosomal peptide synthases, and these are likely to encode secondary metabolic clusters. And this kind of makes me think of the Brasilia-cardin situation in Nocardia brasiliensis. Perhaps these organisms, which are known to produce lots of, or Streptomyces in general, are known to produce lots of immunomodulatory secondary metabolites. So perhaps one of the ways that somaliensis is able to cause disease is through the production of these kinds of molecules to try and reduce the effect of the host immune system. There are horizontally acquired regions within the genome, so I alluded to this before with the bacteriophage story. We tend to spot these through changes in GC content and the association with insertion elements. So this tells us that the genome is plastic, and these organisms are likely to be exchanging genetic material within the environment, either in the soil or with the host. And interestingly, we've never been able to see Streptomyces somaliensis sporulate in solid culture. Streptomyces are largely known to be sporulating organisms, yet we've never been able to see this organism sporulate. However, within the genome sequence, we've identified some genes that appear to be involved in sporulation. So maybe we haven't found the conditions for these organisms to sporulate yet. So that there's some level of investigation that we can do there. And the reason that sporulation is important is these are likely to be the reasons that these organisms can live and survive within the soil. Okay. One slight caveat to this is that the white B genes are iron sulfur cluster containing proteins, so they're likely to respond to oxidative stress. And they're also known that homologs of these are virulence factors in Mycobacterium. So maybe we're not looking at a sporulation gene, we're actually looking at a virulence factor. Either way, it's very interesting from the biology point of view. So overall, just comparing a few Streptomyces genomes, in terms of the overall size of these genomes, you can see the variation in size. Somaliensis is smaller, has a higher GC content. We don't yet know how many protein coding genes there are. There appear to be a large number of tRNA genes, probably more than there are in the other Streptomyces, and yet as yet we don't know the codon density within the genome. So we can align these genomes with known Streptomyces genomes, and you can see that largely there is a clustering of these open reading frames of Streptomyces somaliensis on the top, and they largely fall within the core region of the Streptomyces coelicolor genome. So if we look at the coelicolor genome in more depth compared to somaliensis, what you see is a core region of the genome indicated in dark blue along the bottom, and you see these light blue arm regions, and these light blue arm regions are largely dispensable for growth in culture, and they encode a lot of carbon catabolic systems that allow these organisms to survive better in soil. So as an adaptation, as a pathogen, it looks like it's these arm regions that are actually missing in Streptomyces somaliensis. So perhaps this adaptation to living in the host, they've been able to dispense with a lot of these genes that are involved in soil survival on a larger scale. So the reduced genome fits with pathogenicity. There are known virulence factors there, but there are probably others. We need the comparisons with the other pathogenic Streptomyces to really dissect out what are peculiar to Sudanensis, somaliensis, the new isolates, and try and get to grips with why these organisms cause different diseases in terms of mycetoma or very aggressive mycetoma, as Ahmed alluded to the other day. What genes are important for survival, whether this be in the soil or, as we've heard over the last few days, maybe there's an intermediate host, and I think the genome will give us some clues to this. Detailed investigations of these genomic islands, so trying to identify potentially horizontally transferred regions that could encode virulence factors will be very important. But I think the real important thing at the clinical level for the genome is it allows us to find specific sequences within the Streptomyces somaliensis genome, which will allow us to develop really good, accurate PCR diagnostic methods to look at these organisms and identify very rapidly which organism is causing the disease, so we can expedite treatment and try and understand how these diseases are occurring. So I'd like to quickly summarize that I think there's a need to develop molecular tools and better diagnostics. We need to understand the biology of these organisms much better. And I think comparative genomics offers us a really big advantage with these organisms, because there's a lot of other sequencing data out there for related organisms. And I think also that the next gen sequencing technology will allow us to open up for very new techniques like RNA sequencing, where you can make a copy DNA library from the RNA from your organism, and we can look at how highly expressed certain genes are within the host. So that will enable us to look at organisms that have been isolated from a host and those grown in culture, and potentially identify genes that are upregulated when they cause infections, and then ChIP-seq, which enables us to look at regulators. So I'd just like to thank my collaborators on all of these projects. So Mikkel Kochan and Louise McGibbon were responsible for a lot of the Nocardia work. Alan Harvey and Carol Clements at the Strathclyde Innovations in Drug Research became very interested in mycetoma and nocardiosis when I gave a talk, and they offered all of that screening and the subsequent screening for free as part of the project, so we're indebted to those. To Mike Goodfellow, who's been a long-standing mentor and collaborator, Ralph Kirby at Yang Ming, for spotting the opportunity to be able to sequence this genome, and we wrote the grant, and amazingly, despite there not being much incidence of mycetoma in Taiwan that we know of, the Taiwanese government went for the grant. Professor Jolanta Czerwinska at the University of Wrocław in Poland has done a lot of work with Mike over the years and was responsible for providing a lot of the strains. And then, of course, to Ahmed and to Mohammed and Mogahid, who's in the audience, for my collaborations and getting me over to Sudan. And finally, to Luis Angel Maldonado at UNAM in Mexico for our Mexican strains of Nocardia. So I'd like to thank you, and I'm happy to take any questions. I hope I didn't run massively over time.
Thank you, Dr. Hoskisson, for this elaborate talk. And if people want to take some time from the coffee break, we are happy to take one or two questions. If there is a burning question, let us have it now.
Oliveira?
Thank you very much. You still talk of Nocardia farcinica as Nocardia, and it was previously classified as Mycobacterium carcinogens, according to the mycolic acid content of which is similar to the mycobacteria. Why do you still keep this classification? And do you think it is that the mycolic acid is still although the advancement of molecular biology has taken over many things. Thank you very much.
I think first, sorry, karaoke moment again. So Nocardia falcinica is definitely a bona fide Nocardia based on 16S ribosomal RNA sequencing. It's well established. Way back in the literature, I'm not even convinced. I think Mike could probably answer the historical side of that. But in terms of the mycolic acids are a very important, not only taxonomic point in terms of classifying these organisms, but also a very important functional component, which allows us not only to look at the comparative genomics of all of the mycolata, but I think it allows us to add resolution because these organisms can produce different kinds of mycolic acids. So TB produces different mycolic acids to bovis, to the Nocardias, to the Corynebacteria. We can use mycolic acids as a way of classifying these organisms, but I still think it's valuable even though we have 16S, because 16S, as I showed on the tree, is not as valuable as with Nocardias as it is with many other organisms. I think Mike might want to make a comment on the Nocardia.
The Nocardia falcinica strain that Paul was talking about is the type strain. That's the strain to which the name was attached. And that, I think, was isolated from Guadeloupe by Trevisan, a French scientist, round about the turn of the century, just before. So it was isolated from a case of bovine farcy, or allegedly from a case of bovine farcy in Guadeloupe. I can well understand how people get confused between Nocardia falcinica and Mycobacterium falcinogenes, because some of the strains that were subsequently classified as Nocardia falcinica, when we developed rather better methods of analyzing mycolic acids, as Paul's saying, many of these Nocardia falcinica strains were found to produce multi-spot mycolic acid patterns on simple thin layer chromatography, and that is indicative of Mycobacterium. So Mycobacterium falcinogenes, to come to the point, is a very different organism from Nocardia falcinica.
So falcinogenes is the fistula withers of donkeys?
No, Mycobacterium falcinogenes, I mean, it's Mohammed Hamid, who sadly is not here, who's the expert on this, but that actually causes disease in cattle here, in a very serious disease of cattle, in zebu cattle here in the Sudan.
We'll take a question from Professor Welch.
There is a comment and a question. We began a study about immunomodulating substances, products from Nocardia brasiliensis after we reviewed the Brasilicardin paper. And so we started a 6-year program that just ended a few months ago. We didn't find in any Mexican strain from different strains in Mexico of Nocardia brasiliensis any immunomodulating factor.
Really?
And then I told my collaborator, I am his student now, Lucio Vera, my former student, now is my professor. And we found that I told him, I think that they missed that strain is not Nocardia brasiliensis. They related about a terpenoid type of compound that had a very strong immunomodulating effect, like more than one of the most important immunomodulators substance that we know at present time. And so I said maybe they miss identifying the correct strain. Lucio said, no, how you can say that? That is impossible. They have all the technology, etc., etc., etc. So the thing is that we didn't find it. We asked them to identify our strains. They didn't find them, neither the immunomodulating substance. We asked them for a control reference, and they sent us, and they say any species. Nocardia species. They didn't say Nocardia brasiliensis. So then later on, I don't know if you saw that paper, they reported a new Nocardia named Nocardia terpenica.
Yeah, absolutely, yeah.
Which I believe that is what they found. They isolated. It was not maybe it was a a variant of Nocardia brasiliensis. But anyway, we didn't identify it in any of the pathogenic strains that we analyze in Mexico.
I guess that highlights the fact that we really need to know what strains we're dealing with. So the identification and classification of these organisms is really of paramount importance.
Yes, the lady.
Salman Makboul, Veterinary Research Laboratory. The organism Streptomyces, somaliensis, which you have sequenced, maybe it had some mutation because it was easy to be cultured in the brain-heart infusion body, brain-heart infusion
Yeah, that
media.
works for all of the strains that we have.
Okay, and also it is not sporulated, as you have.
So, I mean, a lot of these isolates, as far as I'm aware, from Ahmed, is that they don't see them sporulating anyway, even when they come freshly out of patients. So one of the reasons that we chose to do the type strain, which we know has been in culture for a long time, is that then we have a comparison against the newly isolated clinical sample, so we can then compare those genomes. So until we've got those sequences, it's difficult to infer whether the mutation is there. And I think we need, as far as I'm aware, nobody's isolated these organisms from soil. So unless we can isolate these organisms from soil, we really need to be able to see what the soil organism looks like that is somaliensis, if there is a soil form of it.
It is not. Is it not easier to take the DNA directly from the patient, and then you?
You probably wouldn't get
Sequence
enough
some
to be able to
markers.
do a whole genome sequence from
Yes.
a single patient.
Yes.
So you would need to passage it at least a few times in culture. Smogahib's got a question.
Thanks, Paul. First of all, let me congratulate you for this innovation and huge work
Thank you.
project, I mean. And if I return back to Nocardia Club which held at Lyon, during that time I think Mike escaped to Shanghai. And I asked P-Man, it's a matter of complaining. We a little bit we found an ignorance to this pathogen in our country. He says this is a similar scenario in US and as well as Canada.
Yeah.
And even when I defend my PhD during the committee, one of the examiner says that why do you choose this ignorable pathogen in Sudan here? This talk in fact provoked me very much, as well as Muhammad Hamid, and it's a matter of frustration, you can say.
Yeah.
And do you think this huge project, Nocardia, deserves this huge project? And if so, why do you jump to proteome to save your money and time to, functional genomic?
Yeah, I mean, we can certainly jump to doing proteomics, and that is definitely something that we can do. I guess I'm a geneticist at heart, so.
So there's a bias to the.
Yeah, I mean, I think without, you can go to the proteome, but it's very difficult with studying the proteome and then doing the mass spec and fingerprinting each of those proteins without having a genome sequence to then go back to and compare it. So we kind of need to do them in succession. But that will be the plan, is to then do the functional stuff so we can do the proteomics. Obviously, microarrays are kind of old news now, and we'll go straight to RNA sequencing to look at transcriptomics. We also have a very good metabolomics facility in Strathclyde, so we'll be able to grow these organisms potentially in serum and compare the kinds of metabolites that they're producing intracellularly and extracellularly to try and see what's going on in terms of their growth and how they function within the host. But I like your comment about Nocardia. A lot of people have said that to me, why are you working on this? And you speak to clinicians in the UK and you say, do you ever see Nocardia? And you say, oh yeah, we see it quite regularly, but we just put it down as an environmental contaminant and throw it away. And I actually, there's a guy who works at the MRC hospital in Gambia, in West Africa, also said the same thing to me. Oh yeah, we see Nocardia quite frequently from tuberculosis-like diseases when we can't culture TB, but we just think it's an environmental isolate and throw it away. So it's massively neglected across the world, and I think the more we look for these organisms in both the industrialized world and in other parts of the world, we will see many more of these cases. I'm sure. It's a very important organism.
Thank you. Let us have the last question from the other room.
No, I think there's nobody next door. There's a question just here. I keep looking into the light.
Until they come up.
I would like to ask you if there are certain genes that tell Nocardia, yes, we left the soil and now we are in human body. You have to turn off that gene that working in soil, and now turn on that gene would survive us in human body. And what about targeting such a gene as antinocardial gene?
That's the plan, is to try and identify these genes. So the first step is to really develop the genetic technique so we can make gene knockouts and then put that gene knockout strain into an animal, a model animal, and see whether we can stop it causing disease. And then we can then begin to think about targeting those specific genes with specific therapies.
It will be detrimental for Nocardia, I think.
Sorry?
It will be detrimental for Nocardia, I think, I would think. What about housekeeping gene? Is there high homology between our housekeeping gene and Nocardia housekeeping gene?
Between humans and Nocardia?
Yeah.
Yeah, there's a lot of difference.
High homology or a lot of difference?
No, there's a lot of difference between them, so that it's very low homology.
I
use the term bacterial housekeeping genes. I do have a very big problem with the use of the term housekeeping genes, because I don't really think bacteria have housekeeping genes in terms of the way eukaryotic cells have housekeeping genes, because there's so much variation in bacterial gene expression across the life cycle and under different conditions.
Certainly. What about MLST and VMLST? What is the difference between them?
So MLST is, as I showed with the 7 housekeeping sequences. With VMLST, you need to know at least 7 virulence genes. So you then have 14 genes, 7 housekeeping, 7 virulence genes, and then you can add greater resolution to your trees. So VMLST is a very new technique. So it's only really been applied to a few Pseudomonas species, but I think it offers us a lot of potential in terms of adding resolution to this tree.
Thank you so much.
Can we have the question from the second room, the other room?
No.
Nothing? Okay, now we close this session and we go for a short break and come back to the